In this video we will discuss about a term that is transcription replication conflicts, and this topic is also relevant to our next paper that we will discuss in our subsequent videos. And this figure is also taken from that papers that we are going to discuss in our upcoming videos. So what is this transcription replication conflict means? So in the DNA, as we know that DNA is present within the nucleus, and in order for this DNA to be converted into the protein, it has to go through this central dogma process. That is, DNA first converted into the mRNA, which is known as transcription, and after that this mRNA is converted into the protein, which is known as translation. And however, one more thing that is parallelly going on within the cells is that replication of the cells. So during the replication of the cells, the DNA also needs to be copied, and these things are usually assisted by this poly PCNA, that is polynuclear. I'm not able to recall its full name, but it's something that helps in the DNA replications along with this DNA polymerase. And what happens is that in the case of tumor cells, there is more TRC has been found, especially in the pancreatic cancer cells. And one of the reagent that researchers has find out that can enhance this kind of conflicts, that is transcription replication conflicts, is this termed as AOH 1996. And what it does is it will leads to the enhanced interaction of this RNAP2 and PCNA, that can be seen in the bottom figure when this AOH 1996 has been induced into the cells. And these frequent interactions will cause this RNAP2 degradations, and at the same time it will also cause the further DNA damage within the cells. That might the DNA damage might be lethal and even leads to the death of the tumors. And the figure of this, or the chemical structure of this AOH 1996, is shown here. And in the paper, what research has shown that their reagent, that is AOH 1996, will cause decrease in the tumors, both in the in vitro conditions and also in the in vivo conditions. And they have checked the patient derived xenograft samples in the mice models, and in that case also they have seen a decrease in the tumors by increasing this TRC within the tumor cells. And this is a graphical representation where tumors of the pancreas can be targeted tumors within the pancreas can be targeted using this TRC, or tumors of the pancreas can be targeted in a way that this TRC will be selectively upregulated in this tumor cells but not in the healthy cells, and that will result in this kind of outcome where the tumor size is shown to be decreased. And however, we will discuss how selective their approach is, in a way that it can target the tumor cells but minimize their impact on the healthy cells. And yeah, I am able to remember this PCNA. It's polycell nuclear antigen. It usually helps in the replication of the DNA along with this DNA polymerase. It is shown in this figure. And when the AOH 1996 is introduced, the interactions of this PCNA and RNAP2 increased, and that might cause some lethal DNA damage that triggers the death of the tumor cells. This is what they seen in the paper This is what they reported in the paper about the in vitro results and in vivo results that we will cover in our subsequent videos. So I hope you understood this topic, that is transcription replication conflict, in brief, so that you will be able to understand our further discussions about the figures that are presented in the paper in details or in a better manner. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.